Well, since you all got quiet right at 2 o'clock, I think I'll start. You know, this is a really special day, and I'm, you know, as I go through this, you know, I think those of you who've joined us now at 2 o'clock, you'll discover that there's about a third of the room who've been with us all day, and we'll give you a background on why they've been here. But let me begin with a few general comments about rare diseases and Rare Disease Day. You know, this movement was founded back in 2008. It's really mushrooms. It's celebrated in more than 100 different countries, and we've been doing it here. And I should have introduced myself. I'm Steve Walkley, director of the Rose F. Kennedy Intellectual and Developmental Disabilities Research Center, which we shortened to IDDRC. The IDDRC has been sponsoring Rare Disease Day since 2012. It's not that all rare diseases are involved with intellectual disability, but a lot of them are, and it's something of real interest to us. There are scattered seats around. Dean Tomaselli, I see you standing in the back, sorry. I started right on time because everybody happened to get quiet at 2 o'clock. There are a few seats on the front row as well, except you won't be able to see me if you sit there, but you know who I am. I'm tall enough, maybe you'll see me. So the IDDRC has been sponsoring Rare Disease Day since 2012. So a key purpose of this day, of course, is to make everyone more aware of rare diseases and of the importance of them in our society and in our medical system and so forth. But it's really also a day to celebrate families that are experiencing rare diseases, and to celebrate them as individuals, both, you know, as mothers and fathers, as well as the kids. And I think you'll recognize, again, the importance of that as we go through the day. According to NIH, there are 7,000 rare diseases defined by NIH and other entities as, you know, affecting fewer than 1 in 2,000 individuals, or fewer than 200,000 individuals in the country. But, you know, the reality is most rare diseases, they're ultra rare. Sometimes we're talking about maybe only 20 families in the country that are identified that have that particular condition in their family. So it's really deceiving to think about 200,000 Americans. That's generally not the case at all. And there are new rare diseases being discovered literally every week. This largely grows out of the new genetic testing procedures like whole genome sequencing. And here and at Einstein, Montefiore, and elsewhere, there are new genes being recognized, mutations being recognized in genes that are contributing to these disorders. And as I mentioned before, many of these cause intellectual disability. And too frequently, you know, it's wonderful, and you know, families can speak to this. I think generally it's good to learn what the cause of the condition is, but that's, you know, after you get over that feeling of, at least now I know what the cause is, you want to do something about it. And too frequently, we don't understand these diseases. We don't understand the pathogenesis of them. And without that understanding, it's very, very difficult to engage in therapy development. So we hope today will help engage you and you know, both the families in the room as well as the scientists and clinicians in this quest for assisting and developing therapies for these rare diseases. So with the IDDRC, we developed a program back in 2017. We collaborated with the Department of Pediatrics. We call it Operation IDD Gene Team. And I just want to spend a minute, because it's the basis of one of the families who's here today, but it's really a major task that we've undertaken in the Kennedy Center IDDRC program to first identify kids that have intellectual disability or autism or seizure conditions and identify the mutation in the gene that is causing that. And once we have that, information, you can see the flow chart here. That patient identification occurs. A lot of times now we're using a program that Melissa Wasserstein, who you'll hear from a little bit later, New York Kids Seek, a U54 grant that funds a lot of this whole genome sequencing that's identifying these mutations. And once we have that gene, Melissa or Bob Marion or others send me that information as director of the IDDRC. We put together a write-up on the gene and the protein and what's known about the condition, and I send it out to the 100 members of the Rose F. Kennedy IDDRC, and looking for a scientist who's working on that gene or that pathway or is interested in doing that. And then we, as a result of that, and these are the genes actually that have been sent out to date, and the ones that are underlined are the ones where we've created these gene teams, which consist of the family, it consists of their clinician, and of a scientist working on that. We get together in the Kennedy Center in the IDDRC main office, and we hold a lay tutorial for the family to help them understand more about what it means to have a diagnosis based on a mutation in that particular gene. And then in some cases we're able to fund with pilot grants made available to us through Dean Tomaselli's office. Actually, we're able to generate some funds to kickstart some projects in these labs. And one really surprising family and one surprising development, and the family here that was part of this is actually in the audience, even though I'm not sure where they are. It's a large audience. Oh, Jessica isn't waving to me in the back. One of these tutorials actually led to the family developing a foundation for their ultra-rare disease. The disease is called SALA disease. Some of you might remember Jessica and Mike. They participated in Rare Disease Day, I think 2 years ago, but then went off and developed their own foundation, which is now supporting research, both with a small grant to Costa de Brenes here at Einstein, as well as to a grant to Bill Gall at NIH. So here's an outline of today's program. And you picked up program cards on the way in, and we're going to, thank you, Kevin. I'd like to introduce Kevin Fisher, who has been instrumental in making today work. Thank you. So we had, just to say one comment here. So we have 3 families that are going to be interviewed, and there'll be a scientist, clinician, and the family up front and telling their stories. And one of these, something very unusual happened here, and I just want to make a comment. This is the Roble family who engaged with MSTP student in Julie Seacombe's lab, because they're working on the gene that's involved with this family. And during that engagement between Hayden and Amy Roble, something really magic happened, in that all of the other Facebook families that were involved with that the Robles knew engaged in this as well. And when they learned that we were going to have the Roble family as one of our families up today in this meeting, all of these other families wanted to come. And so sitting among you are 12 families from all over the US, one from the UK, that all have within their families a variant in the KDM5C gene that leads to a condition in one of their kids. And so we had a whole morning program just for them. And critical to that whole effort not only was Hayden Hatch, but also the Children's Evaluation and Rehabilitation Center with, and I'm going to ask Amy Schumer to come down and just give us a little bit of a flavor of what happened during that program this morning. There's your first slide.
So hi, I'm Dr. Lisa Schulman, and I'm a developmental pediatrician and interim director of the Rose F. Kennedy Children's Evaluation and Rehabilitation Center. And you know, there are magic moments in life. I'm saying this as a kind of behaviorist when there are unique opportunities to learn or accomplish something. Everything's in a row, everything's lined up, and that's what Rare Disease Day is. It's pulling together the family, clinicians, and scientists who are needed to take a step forward. So you know you're not going to make it through today's event without hearing the analogy of the blind men and an elephant parable. So I'll just jump right in with it. Here you see an elephant. Here you see 5 blind men feeling different parts of the elephant. Basically, the analogy is that blind men who discover an elephant, and since the men have never encountered an elephant, they grope about seeking to understand and describe this brand new phenomenon. One grasps the trunk and concludes it's a snake. Another explores one of the elephant's legs and describes it as a tree. A third finds the elephant's tail and announces that it is a rope. And the fourth thinks it's a wall. Each in his blindness is describing the same thing, an elephant, but each describes that same thing in a radically different way. We have a bunch of amazing clinicians here, scientists, and parents who are just awe-inspiring. And we're each at times blinded by our passions, our interests, and our areas of expertise, functioning in our individual silos. And Rare Disease Day is an opportunity to step outside those silos and those passions and talk and come up with a common agenda. And you're going to be hearing about 3 scenarios where that has occurred. And this morning was really quite amazing. Hayden will give you the details, but to me, it was inspirational and basically the meaning of translational medicine, combining resources, expertise, and techniques. And we'll take another step forward during today's program by really focusing on the science. When it comes to rare diseases, there are over 300 million people living with a rare disease worldwide. And 72% of those rare diseases are genetic. And 70% of those genetic rare diseases start in childhood. It's a pediatric issue, and I'm a pediatrician. You got me. The main objective of Rare Disease Day is to raise awareness with policymakers and the public of rare disease, and to reinforce the importance as a public health policy. And programs like this can contribute and give the ammunition we need to do just that. I love this quote from Rare Disease Day a few years ago. You really see and understand the notion that alone we are rare, but together we are strong. So today, again, is an event in the true spirit of Rare Disease Day, a family, clinician, scientist partnership. I am honored, thrilled to be here and be a part of this day. The morning was inspirational. And I wanted to say that we have a multidisciplinary team of clinicians from the Rose F. Kennedy Center, kind of playing part of that clinical role along with Dr. Wasserstein and Dr. Marion. We have physical and occupational therapists leading a children's therapeutic program. We very much enjoyed participating in creating a context for the 12 families that are here. And again, this is one of those magic, rare, teachable moments, and I'm excited to be a part of it. Thank you.
Lisa, thank you. So we're going to start the program. Actually, the very first family is one where I'm going to be the scientist talking to you, and I'm going to spend a few minutes giving you a little background, and then the family will come up, and Dr. Bob Marion will come up to interview them. But let me begin by introducing you to three remarkable young women, Amelia, Celia, and Sarah. They all have a very rare disease called a lysosomal disease. I'll say a little bit more about the lysosome in a minute. It's called alpha-mannosidosis. And pictures of the mom, children, their sisters, here on vacation in Norway. And here's a picture of Sarah and her parents in their home in New Jersey when I visited recently. But before I get into alpha-mannosidosis specifically, to say a word about lysosomes, lysosomal diseases. The lysosome was discovered by Christian de Duve back in 1955. He won the Nobel Prize for that incredible discovery, 1974. But, you know, this was a discovery. It was very close to the Einstein community because we had our own Alex Novikov here at the time, who was the very first person to ever actually see a lysosome and know that it was a lysosome. And he did that with electron microscopy and special markers. De Duve won the Nobel Prize. Unfortunately, Alex Novikov did not join him in that prize. But nonetheless, you know, Novikov was really a major cell biologist on the faculty here for many years. And we didn't, in those early days, we knew almost nothing about the lysosome. But today, we know a great deal. Not everything we need to know, but we know a great deal. We know it's a little organelle in the cell. It contains lots of enzymes because it's a recycling center for the cell. It's where complex molecules go to be degraded and then recycled within the cell. Literally every molecule in cells has to be recycled at some point. And one of those enzymes within the lysosome is alpha-mannosidase. And when that gene is mutated and this protein is missing, we have a disease called alpha-mannosidosis. It was first described back in 1967. It took 30 years before the gene was cloned, but to date, there have been something like 150 mutations that have been identified as causing that disease. But the incidence, this is an ultra-rare disease, like so many lysosomal diseases are and other rare diseases. It's 1 in a million, 1 in a million. And in this case, the missing enzyme is involved in degrading these complex oligosaccharides, very specific mannose linkages within these complex molecules. And when the enzyme is not there, those molecules can't be recycled. And so they build up inside the lysosome. And so, like other lysosomal diseases, there's storage that builds up inside the cells. And in this case, it's these mannose-rich oligosaccharides, glycoproteins, and also some simple gangliosides. There are 3 clinical phenotypes to recognize. Bob Marion actually will say a little bit more about that later. And then a whole cascade of neurodevelopmental events that occur in an individual with this disease. Some of the patients, some of the affected individuals can live into their 4th decade, but there's a slow downhill process involved in that. And just to give you an idea of what's going on in the brain, because we know a lot about that, here's an example of the storage in cells. You really can't appreciate this perhaps, but this neuron is undergoing storage as well. The vacuoles here are filled with these water-soluble oligosaccharides, and so this is what it looks like at the EM level. Other neurons right nearby, this is in the brain, are filled with not only those oligosaccharides, but also gangliosides that are secondarily building up in the cell. We know that some of these gangliosides are like the Tay-Sachs ganglioside or GM2 ganglioside. We know that neurons that store that ganglioside sprout new ectopic dendrites out of their axon hillocks, an unprecedented event for any neurological disease, only happening in lysosomal diseases. These new dendritic processes have new synapses on them. And this is all akin to what Dom Purpura described here at Einstein back 30 years ago, called ectopic dendritogenesis that he discovered in Tay-Sachs disease. So the equivalent occurs in this secondarily storing GM2 ganglioside disease. And in addition to that kind of ectopic dendrite growth, there also are axonal abnormalities, particularly affecting the GABAergic or inhibitory neurons in brain regions like the cerebral cortex and elsewhere. These spheroids can be labeled in cells. We know they look very different at the EM level than what we see in the cell bodies, really reflecting axoplasmic transport defects. I know I'm going quickly, but just to give you a flavor, and when these spheroids form, it appears to lead to the compromise in these cells. These are Purkinje cells in the cerebellum, and these cells start dropping out. They start dying as a result of these large swellings that are occurring down in their axons. So this just gives you a little bit of a feeling for what we know about the progression of this disease. Now let me introduce you to these individuals that I mentioned before. This is Silji, and by the way, I know the Mom family very, very well, met them many years ago, and they've given me permission to talk about their family in some detail here today with you. So Silji was diagnosed in 1983. She developed hydrocephalus in her first year, and then soon after started missing developmental milestones, had intellectual delay, had hearing loss. She was diagnosed when she was 6 years old. This is from her father, Dog. Silji could run a bit clumsily as a young child as she walked 2 kilometers to school by herself in the first years, but not after age 10 because she would fall. The cerebellum with those Purkinje cells that were dying was, I'm sure, contributing to this. She skied downhill mostly without falling until age 12. But then since age 15, she's had yearly psychotic episodes. After age 20, she needed some support. This is the typical picture of many, many individuals with alpha-mannosidosis. From age 30, she's been wheelchair dependent, and she recently had to have some major surgical excisions because of recurrent muscle infection. And so today, according to her father, Dog Mom, Silji is now 37 years old. She's living in a home with assistance 24/7. You think you get a sense of the long-term nature and downhill course of this disease. But now meet her sister, Amelia. She was born just a few years after Silji. She also had null levels of the alpha-mannosidase enzyme, and she experienced very similar early disease. However, Amelia ended up finishing school. She's now 33 years old. Her hearing is near normal. She has no significant motor problems, and she's had no psychotic breaks at all. Today, she works in a kindergarten that's sponsored by the Norwegian government. They live in Tromsø, just inside the Arctic Circle, by the way. She lives independently in her own flat with her dog and her boyfriend. As her father said, she's very active on Facebook and likes to party and to fish and to ski. So I like to say, well, she's a typical Norwegian, right? So why is her course so different? Well, it turns out that reason has to do with the fact that she had a bone marrow transplant in 1997 when she was 10 years old. And the year before this, I visited, my wife and I actually both visited the Mom family in Tromsø. And because 2 years earlier, I published a study using a cat model, a genetic cat model of alpha-mannosidosis to show that in that particular disease, a bone marrow transplant was remarkably effective. And I'm going to walk you through a little bit of that story. So the backstory on bone marrow transplants, they were being done as early as the 1980s, mostly in kids with leukemia. There was a group in London that was doing a lot of this work, and they decided to treat some Hurler patients in mucopolysaccharidosis type 1, another lysosomal disease. However, there was no real sense of why a bone marrow transplant would be helpful in a brain disease like Hurler disease. And so at the time here at Einstein, this was back before the days when we all had mouse models, because this was before, in the late 80s and early 90s, when if you wanted an animal model of a human disease, you really had to go find it in the companion animal population. And so I happened to have here at Einstein during that time feline models of Sandhoff disease, which is like Sandhoff disease, like Tay-Sachs disease. It's a GM2 ganglioside storage disease, and also cats with alpha-manocytosis. These were genetic conditions. We would breed the heterozygotes, generate litters that would have a 25% chance of having kittens that would be affected by the disease. And we undertook a collaboration with a veterinary school at Colorado State University because they were doing bone marrow transplants in cats. And so we transplanted cats with these two diseases, not because I thought it would work, but because I thought, quite honestly, we could at least define the limits of how well it might work. And so I'll tell you the Sandhoff story first. And it was that there was no clinical improvement at all in the cats that were transplanted. Here's what the brain looked like after the transplant. This is a stain for the ganglioside that's building up inside the neurons. The neurons were still loaded with storage. And yet, when we analyzed the brain for the missing enzyme, in this case it's called beta-hexosaminidase, we found that 30% of normal activity was there, but the brain was still diseased. And fortunately, there was a histochemical stain for beta-hexosaminidase, which we did, and this is what a normal neuron looks like in a cat's brain or a human brain. The neurons are blue here because they're loaded with that enzyme and all of their lysosomes throughout the cytoplasm. Here's what it looked like after the bone marrow transplant. I'm not showing you the mutant, but it would be a completely negative stain. But you see lots of blue cells, including some, I think you can appreciate, lining a blood vessel here. These are perivascular microglia now, and then other cells in the parenchyma, sitting in some cases right adjacent to neurons that are, you can just barely see because of a light safranin counterstain that's been applied to them. But there's no exchange between the microglial cell that's got the enzyme and the neuron that's got the storage. And so no correction occurred. Well, we did the same study exactly in the cats with alpha-manocytosis. In this case, the enzyme levels in brain were not quite as high as that 30% that we saw for beta-hexosaminidase. It was like 14, 15% on different parts of the brain. And once again, fortunately, we had a histochemical stain where we could actually see the presence of that enzyme activity. Here's a mutant neuron that's safranin stain, counterstain. So this is just showing you the neuron. It has no blue stain at all because it doesn't generate that enzyme. Here's a normal neuron from a normal cat. After the bone marrow transplant, once again, we saw very positive cells scattered around. Some of them were clearly neurons by their shape, and some cells were very positively stained like this, but others was much more subtle, like shown here, with just a few little blue dots inside what was identified as a neuron. And so, and what was even more important, of course, was as these cats aged out, they didn't develop disease. Even when the untreated mutant kittens had reached end stage, the ones that had been given bone marrow transplants were completely normal. And here's an untreated mutant full of the storage that I showed in an earlier slide. And here's a neuron in the bone marrow treated cat where the cytoplasm is completely normal. And so why does this work? I mean, back in the 90s, there were a lot of different ideas about how something like this could work. A lot of people were still skeptical even after we developed these data, but the idea that is now widely accepted is that when you do the bone marrow transplant, you're of course replacing the whole blood system. That's both the red blood cell system as well as the white blood cell system. And there are cells in the white blood cell population called monocytes that can invade the brain. In fact, they do this in early development in all of us to create the microglial population that's resident in our brains. So when you do a bone marrow transplant, you're replacing some of these cells. When they're there, they act like good Trojan horses where they can secrete that enzyme and and cross-correct the neuron, which can then take it up by different kinds of receptor mechanisms. And just as the Duve predicted, a Nobel Prize winner predicted, that once that protein is on the surface of the cell and is endocytosed into the cell, it ends up being trafficked right to the lysosome, exactly where it's needed. So it's really a remarkable cross-correction that normalizes cell neurons throughout the brain in these experiments with the cats. The other thing that we did, and this was done by Costa de Brenes, who is a faculty member here at Einstein, is he put cat microglia in culture and asked the simple question of, well, do they secrete lysosomal enzymes? And if so, do they secrete some more than others? And if you can read this, you'll see that the secretion is by far the greatest for this one lysosomal enzyme. All of the others are much, much less. And this is really a predictive statement about the value of bone marrow transplantation for lysosomal diseases, where we know today that it does not work for almost all of the lysosomal diseases, but it does work for alpha-melanocytosis. So maybe not appreciated at the time, but bone marrow transplantation probably for alpha-melanocytosis may have really represented one of the first successful treatments of a genetic brain disease since the discovery of dietary therapy for PKU, which I think many of you would be familiar with. So that's the story I want to tell you. I certainly want to thank the moms for allowing me to reveal information about their family. Thank the Forsmans for being here, who you're going to meet in just a minute. Also thank Costa for his efforts in this study. Also Don Siegel, who was here at the time on the faculty as well. And then to make one other little comment about the sort of pathway to the story that you're going to hear from the Forsman family. You know, back in 1995, you know, I mentioned meeting Dog Mom really very early in this. He's a physician as well as a parent of two daughters with alpha-melanocytosis. He was fiddling around, you know, online back in those days, and he met Paul Murphy, also a father of a daughter with alpha-melanocytosis. Her name is Taryn. They've actually been here and spoken to the medical school class before. And they did this, the two of them did this through a new search engine. And those of you who are as old as I am, and if you're web savvy, more so than I am, you'll know that Alta Vista was one of the very first search engines. And so they met each other online back in 1995. Then they teamed up with another father from New Zealand who had twins with alpha-melanocytosis. And the teamwork that went together, they, and I'm happy to say it, very pleased to be on their scientific advisory board that helped them with this. They created a foundation called the ISMRD, the International Society or Advocate for Glycoprotein Storage Diseases. And this past summer, that organization celebrated its 20th anniversary. I was very pleased that they invited me to come. And so, so I go and hear the kids that were present at that organization, that meeting in Atlanta. And one of them, this young woman right here, Sarah Forsman, during the break, I'm going out to get some coffee, and here's Sarah and her father, Jeff. They come up to me and they introduce themselves, and Sarah or Jeff, I forget which one of you said it, you know, revealed that Sarah has alpha-melanocytosis. And you'll meet Sarah in a minute. And I was absolutely dumbfounded because I'm looking at Sarah, but I don't see an individual with alpha-melanocytosis. I see a perfectly normal 20-year-old standing in front of me, engaging me in conversation. And, you know, you guys don't know it, but I was like rumbling around in my brain trying to make sense of it all, thinking maybe I misunderstood. Maybe this is the sister of, you know. Anyway, it was a remarkable experience for me 25 years after we published the paper on alpha-melanocytosis.